Hi everyone, welcome to Basic Science Series. I am your host Lokinder Kumar, and today we are going to understand this, that is the structure and function of the bacterial cell. I have constructed this particular structure from scratch and I have discussed everything in this video. I have made all those flagellar structures, nucleoid, inner, outer, and peptidoglycan layer, and I have discussed everything in this video. So I want you to watch this full video and understand the bacterial cell. Alright, without delay, let's start the presentation. Today, what I'm going to do is I'm going to design a bacterial cell. During that process, I'm going to show you what are the different components of the bacterial cell, what are the function of all those components, so that you have a basic idea of the structure and the function of the bacterial cell. All right. Okay, without delay, now let's start the designing process. Before that, I'm just want to tell you that what I'm right now using is Microsoft PowerPoint. It's a simple presentation software, but there are some options that you can utilize and create the images you want to create. So you can basically make really nice vector images using this softwares. So all right, let's start the designing process of the bacterial cell, right? So since I'm making everything, designing everything in the real time, so it will be a kind of interactive process where you get a chance to learn the function, and you get experience of a podcast at the same time. You will also know how is my designing process, and you may be able to design your own structure. So we're going to continue this series, and we're going to learn a lot of things from this specific kind of interactive process. All right, so okay, before I design, I need to choose the shape of the bacterial cell, right? We all know that bacteria is spherical. That means it's round. It can be round. It can be long, rod-shaped. We call these kind of structures bacilli. So what I'm going to design is a typical E. coli cell. Basically, I'm going to design a bacterial cell which is gram-negative and it's motile. So I'm going to design the structures that are required for the motility, right? So let's first choose the shape. So I'm going to use this rectangular shape. You can change the shape and make it a little capsular structure. So here you are. Great. So now you can see we have a capsular shaped structure. So bacteria usually have a specific shape, right? Why? Because bacteria has cell wall, and eukaryotic cell lack the cell wall structures. So eukaryotic cell has cell membrane. In bacteria, there are cell walls. So in gram-negative, you have gram-negative cell wall. In gram-positive, you have gram-positive cell wall. So there are some molecular differences. There are some basic differences. But right now, we are not dealing with all those differences. But what we are dealing right now is the basic outline of the bacterial cell. So I wanted to show you is what is the basic structure of the bacterial cell, right? So let's start the designing process by making the cell a little larger, so you can see it's nice, and you can change the color. So I'm gonna change its color to a little bit of the dull orange color, and outline, you can choose any outline you want, right? So you can see that it gives you an appearance of a capsular structure. So now what is the next structure that I'm going to design? The next structure is the triple layered gram-negative cell wall, right? So triple layered means you have outer lipid layer, then you have middle peptidoglycan layer, and then you have internal lipid layer. So for that, it's very easy. You can just copy paste this particular structure and you will see that you get the artifact of the cell wall, right? Okay, so you can change the color. So I'm gonna make it little maybe greenish, right? So you can see that the internal compartment is like that. So if you again copy this particular section and change the color of this, so you can see that the appearance of new structure is there. So you can see outer, which is orange color, then you have internal, the middle peptidoglycan layer, and then you have internal layer, and then you need the cytoplasm, right? So basically the idea here is to create the artifact of 3 layer, right? So, and there you are. You can see. So you need to fix these distances so that everything is in proportion, otherwise it looks really bad, right? So now you have the outer lipid layer, then you have internal peptidoglycan, and then you have the middle peptidoglycan, and then you have internal lipid layer again, right? So let's label these components separately. So I'm going to use the line effect. So here I'm not gonna place it on the right side because at this point I'm gonna design flagellum at this particular point. Okay, so this is the outer layer, right? So oops, I moved the main frame. Okay, so let's write, so you can basically incorporate text. Here I'm going to use a specific font. Let's use the Calibri font, and this is our outer lipid. You can call it as a layer, you can call it as a membrane. So outer, let's call it as a layer, right? So you have outer lipid layer. I'm gonna make it bold so you guys can see this particular text easily. And then let's label the second one. I hope you remember that the second one is the peptidoglycan, right? So the peptidoglycan is the middle layer in gram negative, and it's the outer layer in gram positive. So you need to remember these important structures. So we are also going to discuss all these components later in our videos where we're gonna detail out all those components, right? So let's write this as a peptidoglycan. So here you have peptidoglycan middle layer, and then I'm gonna label the third one, which is the inner. So basically change the outer to inner, right? So you have 3-wall, basically 3-layered structure. The first one is the outer lipid, and then peptidoglycan, and then you have inner lipid layer. So that is the basic outline of the bacterial cell. In this case, I'm designing a gram negative cell. So in this case, we are also going to design a flagellum. So flagellum is the organelle bacterial component or bacterial organelle which helps the bacteria to move, and it's like a rope-like structure it helps the bacteria to move into the aqueous environmental condition, right? So, and there are so many different division of the bacterial cell on the basis of the presence of the flagella. If it is present on the you can say if it is present on the one side, then you can say polar flagellar bacterial cell. If you have the large number of bacterial flagella, so you can call it as a polyflagellate bacteria, right? So I'm going to just design a couple of flagella just to give it a appearance of a motile cell, right? So you can use one of the basic feature available in PowerPoint to provide this artifact. So basically you can see that you can make it as a rope-like structure, and I don't like this bump here, so I'm gonna move it and again design the basic structure. So here you have to be careful, because you can easily mess up this particular parts, because in PowerPoint you don't get that luxury of creating things very easily, right? So here you have the nice flagella. As I mentioned here, it is used for the motility. So it's a rope-like structure, and now I'm going to color this, and I'm going to give it a little dark color, right? So you can see a nice artifact of the bacterial flagella, and then you can copy the same structure and then just maybe move it around, and you have two flagellar structures in this bacterial cell here, right? So you can make it a little smaller, and you can also change the thickness of this particular structure. So right here. And the same way you can design as many as flagella you want, right? Depending upon how many flagella is present in particular bacterial structure. So I'm going to design another flagellum in the center here, and these flagellum, they are basically hair-like structures that help bacteria to move in the aqueous environmental conditions, right? So there is some bad artifact, but anyways, so I'm going to use this because I'm going to color the whole thing. So you have all the flagellar structures right here, the bacterial cell. So basically bacteria is using these structures to move in the aqueous environmental conditions, and they are also used in the movement on the solid surfaces or semi-solid surfaces like media, agar media, and all those things. So flagella have there are various functions that are performed by the flagellum, right? So you have so now right now you have flagella and outer layer, peptidoglycan, and the inner layer. Okay, so I'm going to label the flagellum, right? Okay, so that's the flagellum, one flagellum. So this is the particular structure that helps the bacteria to move under various conditions. All right, so now there are internal organs or internal organelles that are useful for bacteria to basically survive under various conditions, right? It requires proteins, so there should be something that synthesize the proteins inside the bacterial cells, and those are ribosomes. So let's design ribosomes, and ribosomes are spherical structures. So I'm gonna just provide a smaller dot here and there in the bacterial cell to just give it an appearance of the ribosome structures. So the structure of the ribosome is a little complicated than that, but right now we are just trying to understand what are the different components in the bacteria. So I think right now it can do the job, right? So you have this these round shaped structures, they are ribosomes. So let me label this. Okay, so as I mentioned, that these are the structures which are called as ribosomes. And there is a basic difference between the eukaryotic and the prokaryotic ribosomes that we are going to discuss in our new videos. Next videos that I'm gonna discuss the internal structures in detail, right? Okay, so you have ribosomes. So I'm just pointing out one, so I will just say ribosome. Okay, and what are ribosomes? Basically, they are microscopic protein factories. They produce protein in a large quantities that bacteria can basically utilize those proteins to survive, because proteins are really essential for any living organism, right? So these are the basic structures. Let me also move this a little that side. So as you can see, these are the basic components of the bacteria, and we also need to label the cytoplasm. This cyan color area is the cytoplasm. Okay, so let me just change the name. Basically, I'm copying the already labeled these names, and so that you can just rewrite on that. Otherwise, you have to change the font every time. So anyways, so I'm pointing out to this particular area, which is cyan in color, light green kind of color. I'm not pretty sure. I'm not good at naming colors or identifying different colors, but this whole area is the cytoplasm. And the cytoplasm, or it's also called as protoplasm of the cell, which is really essential for cell growth, metabolism, and DNA replication, protein synthesis. Everything is happening inside that compartment. So it has a lot of different things like nutrients, various waste products. They are also generated in the cytoplasm. So anyways, it will be a long discussion on specifically on each structure. Let me tell you one thing, that every structure is a very complicated structure and really important for the bacterial cell, right? But we are not going to explain everything in detail, but we are going to know that what are the basic functions of all these organelles, all these structures. So next thing, what I'm going to design is it will take some time for me to make all those structures, but it is an important part of the bacterial cell, and it is present on the outer layer of the bacteria, and this is called as pili. 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 So pili are basically hair-like structures that are present on the surface of the bacterial cell, and they are tiny, they are small than the flagellum, and the main function of the pili is sometimes they are used as the attachment And during the attachment process, they are also sometimes used in the movement on the solid surface. So there are various functions that pili perform. They are also used to transfer the genetic material from one bacteria to another. So here is the pili, and let's choose a different color, right? Okay, so I'm gonna choose dark color, maybe. Okay, let's choose the black color for that, and they are distributed on this surface basically throughout the surface. So it is going to take some time for me to actually design all these structures. So I'm going to fast forward this particular section of the video. All right. Well, it took a while. It requires a lot of patience to detail out all these small structures. But, the end, you can see the bacteria is looking, it's getting the scary look of the bacteria because of those pili structures. So I'm going to move this peptidoglycan a little away from this so that I can see that this is standing out separately, and you have outer layer, right, and pili structures. Okay, so we have most of the basic structures that are aligned, and now the important part of the bacterial cell, that is the DNA, genomic DNA of the bacteria, right? We all know that DNA, that is the genetic material of the bacteria, is the main source of the information, which is converted into protein form using those ribosomes. So let's make the DNA. It's also known as nucleoid of the bacterial cell. And simply, I'm gonna just make the DNA structure somewhere right here. Usually, we know that DNA is a double strand. DNA has the double strand, and it's a polymeric structure. But right now I'm not going into the detail of the DNA. I'm just making a nucleoid structure, just a long polymeric thread. You will see in various papers, or if you Google search the image of the E. coli DNA, you will see a long like a strand structures that are present in the cytoplasm. You might have seen a picture where E. coli DNA is as shown. It's a SEM picture which is available on internet. You can Google search it. So basically DNA is a long strand. It's made up of nucleotides. So we are not going to discuss all those details, but I'm really having a hard time in designing the possible correct structure for this nucleoid. Okay, so here, well, that looks okay, right? Okay, so we have, so we have this nucleoid structure, and I'm going to place it at right here in the center. So let's change the color to maybe something a little darker. So here, that's the nucleoid. Now I'm going to label this as nucleoid. So let me find space right here. Great. So I'm going to change it to nucleoid. Now you know what is nucleoid. Nucleoid is basically the genetic material of the bacterial cell. Right here, a little too far, but you can see it, right? So, all right, so anything else in the bacterial cell? Maybe we can make some more ribosomes just to cover the emptiness of the cytoplasm, right? Okay, great. So this is our bacterial cell, and you can see all the basic structures of the bacterial cell. So let me summarize again what are the different components. So first, what we have done is we designed the outer compartment of the bacteria. Then we discussed the cell wall, that is gram-negative cell wall in this case, where you have outer layer, then you have peptidoglycan, then you have inner layer. Then we constructed the cytoplasm and we constructed the flagellum. Flagellum is the organelle that is required for the movement of the bacteria from one place to another. Then we designed the ribosomes. Ribosomes, we all know that these are the protein factories that synthesize proteins inside the bacterial cell. Okay. Then we again designed these small hair-like structures. These are Pili structures that basically give the scary appearance of the bacterial cell. Okay, so what was the last structure? Last structure was the nucleoid, which is the DNA, deoxyribonucleic acid, which is required to basically to give the cell the information of the different proteins, because you know that the nucleoid is made up of a nucleotide. It's a sequence of adenine, guanine, cytosine, thymine. Basically, all the genes, they are present inside the nucleoid, and then ribosome convert these genes into protein form. So all the information is present in that nucleoid. It's the central part of the bacterial cell. It's really important, right? So this is your bacteria. So if the next time if someone asks you, okay, what is the structure of the bacterial cell? So I think you should be able to explain the basic structure of the bacterial cell, right? So I hope that the functions were also clear of each component of the bacterial cell. I try to explain the functions. I try to explain the specific structure in this video. So I hope that this whole concept is really, really clear for you, and you completely understand the basic concept of the bacterial cell, right? So we'll meet in the next video where we will discuss different topics. There are so many different topics that I want to discuss. The molecular mechanisms or the structures of the specific organ of the bacterial cell or human cell. There are so many structural components, and there are so many proteins that are involved, right? So using this particular video format, we are going to design all those structures. We are going to understand the function of all those structures in detail, right? So I hope that you're going to like this video, and if you like the video, please hit the like button. And if you want to share this video, you are most welcome to share the video with your friends so that they can also understand the basic concepts of different structures of the bacterial cell, right? So for now, I'm going to just end this video, and I'm going to meet you in the next video presentation where I'll bring more complicated and more interesting topics, including the cellular signaling, which is the most important and complicated part of the bacterial cell, as well as the human cells, right? So we have a lot to discuss and a lot to understand, specifically in the biosciences. Structures are really important, right? Using this technique, we can understand each concept in a kind of nice and interactive, in a fun way. So thank you for watching the video, and I'll see you in the next video. Thank you and take care.